Hi folks, this is Dr. V. Sivaramakrishnan from SRM Institute of Science and Technology. I welcome you all for this presentation on mitochondrial genome, its limited autonomy and genetic disorders. What exactly is mitochondrion? Mitochondrion represents a single mitochondria. Within a cell, there could be multiple mitochondria that could be basically present, and mitochondria are nothing but cytoplasmic organelles which are present in any eukaryotic cell whose main function is to generate a chemical entity which is called the ATP or the adenosine triphosphate, which happens to be the energy currency of the cell. They mediate cellular respiration through the electron transport chain and as well as the entities of the electron transport chain or the coenzymes called as Q and as well as the cytochrome C complex. It has an independent extra chromosomal genomic DNA which differs from the nuclear DNA. Mitochondrial DNA has a key role in maternal inheritance of the genes, and this was first discovered in the latter part of the 19th century. How did the mitochondria originate? This was basically postulated as a theory called as the endosymbiotic theory. We know that mitochondria, which is the human cell counterpart or the eukaryotic cell counterpart, and as well as the plant cell, which also happens to be eukaryotic cell, the counterpart present equivalent to the mitochondria is called as the chloroplast. Both share certain similarities. Both are double membranous and both they have an essential role in energy conversion. Both have their own respective independent genomic DNA and as well as certain number of ribosomal RNAs. Both organelles are believed to have originated from these kind of, hence we believe that it could be due to the endosymbiotic kind of an association. Both mitochondria and chloroplast have proton gradients which enable to actually generate the energy currency of the cell, which is called as the ATP molecules. These organelles were incorporated into early part of the eukaryotic cells through a process which is what we call it as the endosymbiosis. This endosymbiotic theory suggests that all eukaryotic cells they arose from a single common ancestor, and we believe that sequence alignments further also provide support for this endosymbiotic theory, which proposes that early eukaryotic organisms they actually engulfed the bacteria and subsequently became symbiotic with the eukaryotic counterparts rather than being digested. Now let us understand the mitochondrial genome. Mitochondrial DNA is way too smaller rather than the nuclear genome. It is circular as you observe in this particular figure, and it is also considered to be extranuclear. The length of the mitochondrial DNA is as small as 16,569 base pairs only. In 2 to 10 copies are observed per mitochondrion. The base composition predominantly happens to be GC rich. The heavy strands which are present towards the exterior regions, if you have to actually represent it towards the outer side, okay, and the inner part represents the lighter strand which is rich in cytosines. The heavy strands are rich in guanines, and if you look at the total number of genes, there are 22 tRNA encoding genes. There are 2 rRNA genes like the 16S and the 12S rRNA, and there is also the electron transport chain or the protein coding genes which are 13 in number. Put together, there are a total of around 37 genes which are making up the component of the mitochondrial genome. Now let us see the salient details of the mitochondrial genome. It can be divided into 4 sections. The first one for the RNA genes. 24 RNA genes they produce the 2 rRNAs and the 22 tRNAs, and each of these 22 tRNAs are represented here in this figure as thin red bars which are labeled with their single code amino acids. The protein coding genes represent the second level, and wherein you have the 13 genes of the electron transport chain, which are the protein coding genes that are participating in the oxidative phosphorylation responsible for the synthesis of the ATP molecules. It is NADH dehydrogenase subunits, which are from ND1 to ND6 and as well as the ND4L, and ATP synthase subunits, which involves the genes ATP6 and as well as ATP8, cytochrome C oxidase subunits, which is the CO1 to CO3, and the cytochrome B unit. The third represents the set of genes which are responsible for the which act as the promoters and participate in the transcription process. There are two promoters, one each for the heavy strand and as well as the light strand, and they transcribe the H and L strands in opposite directions to actually generate these large multigenic transcripts, and later they are cleaved once they are basically copied. And the replication involves the fourth step, wherein the origin of replication is one each present in the H strand and as well as the L strand, and the dashed arrows, they actually show the DNA synthesis directions. There is also the observance of these small D loop region in addition to what we have spoken about the H strand and the L strand. Please make a note of this. These are the critical salient details of the mitochondrial genome. The important features of the mitochondrial genome includes it doesn't favor any occurrence of the crossing over. There is no DNA repair mechanism, and as I already mentioned, there are only 2 to 10 copies per mitochondria. But however, this will vary from a particular cell type to cell type. Say for example, in case of a spermatozoa, there is just only 100 mitochondria that is basically present. But whereas in the case of a muscle or a cardiac cell, something like that, wherein close to around 1 lakh number of mitochondria basically present, and this tells you the energy need of that particular cell type also. And it leads to a high exposure to the oxygen free radical generation as a byproduct of your ETC electron transport chain, and no histones are basically present here, which since this is just only a small DNA, it doesn't need any kind of a folding or supercoiling to be basically done, and it is very easy to actually hold within a mitochondria, and it doesn't have any kind of intronic regions or the non-coding regions. And the most critical aspect of the mitochondrial genome happens to be the ability for occurrence of maternal inheritance. What does it mean by maternal inheritance? Here, when an affected female only transmits the disease to all their children, whereas the affected males do not transmit the disease to their children, so they actually have only normal children. Males hence cannot transmit the disease as the cytoplasm is inherited only from the mother and the mitochondria are present in the cytoplasm. Now let us see the pedigree that I have actually represented over here. We just check out from an affected female or the mother, only all the children basically are getting the mitochondria, and they all seem to be are said to be affected. Whereas further, if you move on to the next F2 generation 2, you can see that only in the female mother being affected, they are transmitting the disease to all their children, as you can observe towards this particular scenario, and as well as in this particular case scenario 2. And if you take the next level of generation also in the F3 generation, you can also observe that here too both the children are basically affected, and that wherein also it is transmitted from the affected mother. So you can see the level of inheritance invariably coming from actually the mother to the children, particularly the transmission happening from females to the females. Whereas you can also observe that this is also seen, not seen, I'm sorry, in the case of the males invariably. You can actually observe in this particular place wherein though there is only the father who is affected, but all their children are not affected. So this is a characteristic feature of the mitochondrial inheritance, which is purely happening through the maternal component. We can appreciate the mitochondrial genome only when we learn about how it differs from the nuclear genome. If you look at the size of the nuclear genome, it is huge, up to 3.1 gigabytes, whereas the mitochondrial genome is just only 16.6 kb. There are 23 pairs of chromosomes, or the number of different DNA molecules in the case of the nuclear genome, but whereas there is only one circular DNA molecule in the case of the mitochondrial genome. The total number of DNA molecules per cell, it varies in the case of the diploidic cell, which happens to be 46, whereas it is just only 23 in the case of the gametes. Often several thousand copies of the mitochondria is basically seen, and it varies from cell type to cell type, as I already mentioned. The associated proteins, the histone and the non-histone proteins, are critical components of the nuclear genome, which is required for the folding and packing of the DNA component within the nucleus. But whereas no such requirement is needed in the case of the mitochondrial genome, and hence it is actually said to be only one circular and much smaller in size, and there are no histones present in them. The number of protein coding genes go to up to 20,000, and it's only 13 in the number which is the component of the electron transport chain in the case of the mitochondrial genome. Probably around 20,000 RNA genes are basically seen in the case of the nuclear genome, and still more research is going on this particular aspect, because this is still a kind of a conundrum, because we have the other variants of the RNA which we speak about, like the miRNA and the siRNA and other variants of the RNA. And in the case of the mitochondrial genome, there are only 24. We have seen the 2 rRNAs and as well as the 22 tRNA. In the case of the nuclear genome, the gene density is 1 per 80 kb, whereas in the case of the mitochondrial genome, it is just 1 per 0.45 kb. There are up to 15,000 pseudogenes which are observed in the nuclear genome. None is basically observed in the case of the mitochondrial genome. At least 50% of the genome and perhaps 67% happens to be repetitive DNA in the case of the nuclear genome. There is no repetitive DNA basically observed in the mitochondrial genome. The transcription, many genes are independently transcribed in the nuclear genome, and we have already seen the mitochondrial genome being extra chromosomal, and multigenic transcripts are produced from both the DNA strands, which are the H and the L, and the introns, they are basically found in most protein coding genes and many RNA genes in the case of the nuclear genome, whereas it is completely absent in the case of the mitochondrial genome, as I already mentioned. And the amount of protein coding DNA is approximately 1.1% in the nuclear genome, and it's 66% in the case of the mitochondrial genome. There are 61 amino acid codons plus 3 stop codons which actually are seen in the nuclear genome, and there are 60 amino acid codons plus 4 stop codons which are basically observed in the case of the mitochondrial genome. In the case of the nuclear genome, at least once for each pair of homologous the chromosomes at the meiosis stage, the recombination basically can be observed, but this is not at all seen in the case of the mitochondrial genome, as there is no evidence for any crossing over. And inheritance perspective with regard to the nuclear genome is that Mendelian for DNA sequences on the X chromosome and autosomes, paternal for most DNA sequences on the Y chromosome, whereas in the case of the mitochondrial genome, it is exclusively considered to be as maternal. Given that there are 20,000 odd genes which are encoded by the nuclear genome, only 37 of those genes are basically encoded by the mitochondrial genome. Now you would have understood that there is an autonomy that is existing for the mitochondrial genome, whereas it seems to be only limited. Why? Because there is independence in protein synthesis, wherein the mitochondrial genome produces the ribosomal RNAs and tRNAs that are only necessary for their own protein synthesis on the mitochondrial based ribosomes. However, the limitations include the mitochondrial DNA, which specifies only 13 out of the 80 proteins that it basically requires for the oxidative phosphorylation mechanism. It does not encode any of the core proteins that are needed for the purpose of making copies of its DNA, like the replication process or its regulation via the transcription process, and as well as if in case there are any mutations happening in them, there are no repair mechanisms available within the mitochondrial DNA. The third reason is the nuclear gene involvement, where the proteins are required for the mitochondrial protein synthesis machinery, are encoded by the nuclear genes. Over 99% of the mitochondrial proteins, they are encoded by the nuclear genes. And the fourth reason, the protein production and as well as the import. Mitochondrial proteins, they are synthesized on cytoplasmic ribosomes. These proteins are then imported into the mitochondria. Due to these reasons, we can say that there exists limited autonomy of the mitochondrial genome. Mitochondrial DNA is prone to occurrence of more mutations. Whenever the defective gene or the mutation is present in the mitochondrial genome, it affects the energy metabolism, and therefore it affects the tissues that require constant energy support. Like for example, the muscles, predominantly the cardiac muscles or the brain muscles, or invariably also to a little extent the retinal muscles, and also neurons invariably need the presence or the activity of the mitochondria. Whenever the mutations happen, we have listed some of them over here as the mitochondrial genetic based disorders. They are called as the Leber hereditary optic neuropathy. In short, it is represented as LHON, and it basically involves loss of vision, which is observed over here, particularly in the central region of the eyes or the retinal region. The mitochondrial encephalopathy with lactic acidosis and stroke-like episodes, which are represented as the MELAS in short. That is, this particular disorder leads to the occurrence of or it affects the brain due to the encephalomyopathy conditions, and as well as results in causing cardiomyopathy related problems. Kearns-Sayre syndrome or the KSS syndrome, it involves retinopathy and as well as even the ophthalmoplegia kind of a problem in these individuals. And the next one is the chronic progressive external ophthalmoplegia, which is represented as CPEO. Here too you can observe the mitochondrial mutations or the deletions in them. The next one is called as the myoclonic epilepsy with ragged red fibers, wherein the predominant cause is the epilepsy, which also can lead to the occurrence of stroke in these individuals, which is represented as the MERRF in the shorter abbreviation. There is the neurogenic weakness with ataxia and retinitis pigmentosa, represented as the NARP syndrome, or the last one, which is the Leigh syndrome, wherein it is due to a pancreatitis related problem, which results in due to the occurrence of a mutation happening in the mitochondrial genome. Here the mitochondrial DNA disorders happening in the specific clinical phenotypes with their respective kind of mutations seen in which particular gene, and whether are they heteroplasmic or homoplasmic, and the mode of inheritance in the mitochondrial DNA based mutations, or the kind of inheritance particularly with regard to the either it is maternal, predominantly as observed in most of the mitochondrial DNA disorders, and as well as in certain case scenarios is also said to be sporadic by nature, is shown over here. For the set of diseases or the disorders we spoke about earlier. All these information provides you the importance of mitochondrial DNA mutations and the critical role of maternal inheritance in it. I thank everyone for patiently listening and conclude this session.